Greetings of the day, dear students. One of the important instrument in microbiology laboratories, hospital settings and the clinical laboratories are laminar airflow. This is one of the important aspects related with the sanitation aspects of air, and this laminar airflow is one of the fundamental instruments that is required for any microbiological laboratory to perform the inoculations and the laboratory work in a contaminants free environment. And that is the next topic mentioned in the syllabus, module number 5 of Sanitation Microbiology, that is laminar airflow. Laminar airflow, or simply known as laminar flow hood or cabinet, it is an enclosed workstation that is used to create a contamination free work environment through filters to capture all the particles entering into the cabinet. So this is a specifically made cabinet that can be used for variety of sterile aseptic procedures, and that is commonly used in microbiological laboratories. And these cabinets are designed to protect the work from the environment and are most useful for the aseptic distribution of specific media and the plate and all the aseptic work that is happening in the microbiology laboratory. And these laminar flow cabinets are very similar to biosafety cabinet, with the only difference is being that the laminar flow cabinet, the effluent air is drawn into the face of the user. In a biosafety cabinet, both the samples and users are protected, while laminar airflow cabinet, only the sample is protected and not the user. And the special basic level of biosafety cabinet is known as laminar airflow. And this is one of the important aspects of sanitation microbiology, especially to prevent the contamination that is happening in the air through the major type of physical method known as filtration. As you can see here, this is the laminar airflow. You can see the same instrument in our laboratory also, where generally students, they generally don't use this one, but the pouring of the medias by researchers and faculty members, they are using. Even students are using when you are going for the higher level like post graduation and all those studies. So this is the chamber. You can see this researcher is working. Inside this chamber there are medias and there are plates and all those things. You can see this is a sterile cabinet, and where we can in front of this, where we can sit and work, and you can do all inoculation work and media pouring and all those things. You might have seen your teachers are working in some of the occasions. And the major component of laminar airflow hoods are, there should be a cabinet, it should be a workstation, there should be a filter pad or pre-filter, there should be a fan or blower, then there should be a UV lamp, there should be a fluorescent lamp, there should be, most important thing is known as HEPA filter. Now we are going to discuss these things one by one. First one is the cabinet. The cabinet is made up of stainless steel with less or no gaps or join which prevent the entry of the spores. Not only spores, the vegetative cell as well. The cabinets provide insulation in the inner environment which created inside the laminar airflow and protect it from the outside the environment. The front of the cabinet is provided with a glass shield, which is in some laminar cabinet opens entirely, or in some has two openings for the user's hand to enter the cabinet. The next one is the working station. A flat working station is present inside the cabinet for all the processes to be taken place. The culture plates, the burner and loops are all placed on the working station where the operations are taking place. The workstation is made up of, the top of this workstation is made up of again stainless steel which prevent the rusting. Then the next part is the filter pad or pre-filter. A filter pad is present on the top of the cabinet through which the air passes into the cabinet. The filter pad traps dust particles and some microorganisms, especially the spores, from entering the working environment with the cabinet. Then the next one is a fan or a blower. A fan is present below the filter pad that sucks in the air and moves it around the cabinet. The fan also allows the movement of air towards the HEPA filter in which the microorganisms are trapped while passing through the filter. Then the next part is UV light. Most of the laminar airflow hood, they will be having a UV germicidal lamp. You know that they sterilize the interior of the cabinet and content before the operation. So the UV lamp is turned on 15 minutes before the operation to prevent the exposure of UV to the body surface of the user. So whenever you apply the UV rays, the microorganism completely destructed by using UV light. When the UV light penetrates the microorganism, the photochemical oxidation, the nucleic acids will be damaged by the formation of thymine dimer, okay, or pyrimidine dimer, and which cease the metabolism and prevention of the replication of the DNA, and the microorganism will be died. The next one is the fluorescent lamp. A white or a bright colored fluorescent light is placed inside the cabinet to provide ample light during the process, whatever we are doing inside the laminar airflow. So that is a function of a fluorescent lamp. And the most important part of a laminar airflow hood is the HEPA filter, that is high efficiency particulate air filter, which is present within the cabinet that makes the environment more sterile for the operation. And this filters the pre-filtered air pass through the filter which traps fungi, bacteria and dust particles. The HEPA filter ensures the sterile condition inside the cabinet and reducing the chances of contamination. The HEPA filters are mainly composed of a mat of randomly arranged fibers, and these fibers are typically composed of a fiberglass and possess the diameter between 0.5 to 2 micrometer diameter, where microorganism will be trapped, and they consist of a thin pleated sheet of boron silicate microfibers with the aluminium separator. They are particulate filter which retain the airborne particle inside the chamber and that remove the contamination. The major factors which affecting the function of these HEPA filters are the fiber diameter and the thickness of the fiber and the phase velocity of the air. Then the air space between HEPA fibers is typically much greater than 0.3 micrometer. So these HEPA filters that is commonly present here over here that ensures the complete sterility inside the chamber, okay, and that is the structural compartment of a laminar airflow. Now the principle or working of a laminar airflow. The major principle of laminar airflow, that is the laminar airflow, the filtration through the cabinet, laminar flow of air through the cabinet. The device works by the use of inward flow of the air through the one or more number of HEPA filters to create a particulate particulate free environment. All the microorganism, dust particles and particulative materials will be trapped inside the HEPA filter when it is passed through the filter. The air is taken through the filter system and then exhausted across the work surface as a part of laminar flow of the air. The air first passes through the filter pad or the pre-filter that allows a streamlined flow of air into the cabinet. Next, the blower or fan directs to the air towards the HEPA filter. The HEPA filters then trap the bacteria, fungi and other particulative materials so that the air moving out of its particulative free air. Some of the effluent air pass through the perforation present at the bottom of the cabinet, but most of it passes over the working bench while coming out of the cabinets towards the face of the operator. The laminar flow hood is enclosed on the sides and the constant positive air pressure is maintained to prevent the intrusion of the contaminated external air into the cabinet. And the main operation inside the cabinet and the primary mechanisms are filtration, that is mainly sedimentation, electrostatic attraction, interception, inertial impaction and diffusion. These are the primary mechanism along with the filtration. Then what are the procedure for running the laminar airflow cabinet? Before running the laminar airflow cabinet, the cabinet should be first checked thoroughly to ensure that nothing susceptible to UV rays are present inside the chamber. So whatever we are going to do, it should not be susceptible to UV light, and that should be removed before you start processing the laminar airflow chamber. The glass shield of the hood is then closed, completely closed, and the UV light is switched on. The UV light should be kept for approximately 15 minutes to ensure the surface sterilization of the working bench. Then the UV light is switched off and the time period of around 10 minutes is spared before the airflow is switched on. About 5 minutes before the operation begins, the airflow is switched on. The glass shield is then opened and the fluorescent light is also switched on during the procedures, whatever you are doing. To ensure more protection, you can clean the entire surface by using 70% ethanol, and that can be used as a complete sterility along with this other typical sterilization procedure inside the laminar airflow. Once the work is completed, the airflow and the fluorescent lamp both are closed and the glass shield is also closed. Then these are the two types of laminar airflow system. One is vertical laminar airflow cabinet. Next one is horizontal laminar airflow cabinet. As the name indicates, vertical, you can see the air is, the HEPA filter is on the top, and you can see the air is coming from top to bottom, and you can see this is spreading like this. And in the horizontal, the HEPA filter is here and the air is coming towards the user. Okay, and this is the horizontal laminar airflow hood system, where this is the HEPA filter. Okay, this is the fan. Okay, and these are the structural compartment, whatever we have been telling. Okay, the air is passing, and here you can see the air is passing through this direction. Okay, and so vertical, you can see it is coming from the top. Okay, as I said, this is coming from the top and it is moving the HEPA filter. This is a HEPA filter over here. Here the HEPA filter is here. Okay, as you can see in the diagram over here. Now the vertical laminar flow cabinets. In vertical flow cabinets, the air moves from the top of the cabinet directly towards the bottom of the cabinet. Okay, from the top to the bottom of the cabinet. Then the vertical airflow working bench does not require as much depth, and the floor space like a horizontal airflow hood, which makes it more manageable and decreases the chances of airflow obstructions or movement of the contaminated air downstream. The vertical laminar airflow cabinet is also considered as safer, and it doesn't blow the air directly towards the person carrying out the experiment, because the air flow is towards from the top to bottom, and it requires less space, and this very safe to use. And the major merits of vertical laminar airflow are the hood not as deep, it requires less floor space, suitable for compounding sterile product, the safety, air not blowing directly at the operator, and sash provides a barrier in front of the operator face. Then the filter on the top easier to access, less turbulent flow from air striking the large object or processing the equipment, less cross contamination of the item positioned on the work surface. These are the major advantages of vertical laminar airflow cabinet. Then what are the disadvantages? Overhead clearance requirement is the major thing, that is changing the filter or servicing unit may require a step ladder. It's a very, always we need to clean the filter, and you need to service the filter. Periodically the filter need to be changed, and cannot place the items or hand on the top of the other items. We cannot place the items or the hands on the top of other items. Increased turbulent flow of air striking the work surfaces. So that is another important issues. Then comes the horizontal laminar airflow. In the horizontal laminar airflow cabinet, the surrounding air comes from behind the workbench, which is then projected by the blower towards the HEPA filter. The filtered air is then exhausted in the horizontal direction to the workplace environment. One merits of this cabinet is that the airflow parallel to the workplace, which cleans the environment with a constant velocity. The effluent air directly hit the operator, which might reduce the security level of this type of laminar airflow cabinet. Here you can see the direction of air towards the user. Okay, this is the kept here, and here you can see the more space in comparison with this one. Okay, and there are also a lot of merits. Okay, there are also a lot of merits which reduce the turbulent effect of air striking the workbench. Then no sash, easier to work and position the equipments, but air blows directly an operator. Then the hands and gloves are generally less contaminating since they are downstream of the sample. Okay, the hands and the gloves are very less contaminating because they are kept downstream. The disadvantages are the filter changes or service usually require repositioning of the hood for the rear access. Larger sample obstruct the laminar airflow, may contaminate the downstream sample also. The blows, fumes or powders in operator's face is constant problem because the operators are sitting here and the flow is towards the face of the operators. So it creates some blows of fumes and powders to the operator's face. Then what are the major applications of laminar airflow hood is? Laminar flow cabinets are used in laboratories for contamination sensitive process like plant tissue culture. This is very important, and we can use in specifically plant tissue culture because that is one of the important sensitive step like culturing of the plant, explant and callus, etc. So other laboratory procedure like media preparation, media plating, culture inoculations. So whatever microbiology work we are doing in the workbench can be done in the laminar airflow hood. The operations of particular sensitive electronic devices are also permitted inside the cabinet. Okay, the entire instruments, like as you can see in the figure in the beginning. Okay, so we can keep some of the instrument over inside, and we can work that instrument inside the laminar airflow. Then in the pharmaceutical industry, the drug manufacturing techniques, drug preparations are also performed inside the cabinet to ensure a particulate free environment during the operation, especially vaccines and all those things. Nowadays, you know that vaccines are very important at this point of pandemic. So the importance of laminar airflow is very high over here. Whatever the sterile work, whatever we are doing inside the laboratory, that should be done in the laminar airflow. Whatever you are doing, the practical work, all the practical work you are doing in the workbench can be done inside the laminar airflow. So the laminar flow cabinet can be made tailor-made for some specialized work and can also be used in general laboratory technique, both microbiological as well as industrial purpose, especially in industrial microbiology fermentation technique, scaling up the microbial inoculum, and screening the microorganism. All those things can be done inside the laminar airflow. And while we are working with the laminar airflow, there should be adequate precautions. The most importantly, the laminar airflow cabinet should be sterilized with the UV light before and after the operation. That is very, very important. When you use UV light, the UV light is very dangerous and the airflow should not be used at the same time. The UV light should be very carefully treated, and the UV light and the airflow should not be used at the same time. No operation should be carried out when the UV light is switched on. So you should not, do any operation when the UV light is switched on, because UV light is highly carcinogenic and mutagenic substance. The operator should be dressed in the lab coat and with a long gloves. Okay, whenever you work with the laminar airflow, you should wear your apron, the lab coat, and you should wear the hand gloves also. Okay, and you can see instead of this you can wear the head cover also. Okay, so that is another important precaution. Then the working bench, glass shield and other component present inside the cabinet should be sterilized before and after the completion of the work. Before and after the completion of the work, they should be sterilized. And this is one of the important aspects where you need to study. So that's all, dear students, laminar airflow, about laminar airflow in sanitation microbiology as well as the air quality standard monitoring. Okay, that's all. So once again we can glance through the major concepts studied in this particular class. The class begins with a brief introduction of what is laminar airflow chamber and what is the comparison between laminar airflow and biosafety cabinets, and what are the components or structural part of laminar airflow hood, what is the principle or working of laminar airflow, what are the procedure for running the laminar airflow cabinet, what are the different types of laminar airflow, vertical laminar airflow and horizontal laminar airflow, what are their merits and what are their demerits, what are the applications of laminar airflow hood, and what are the precautions while we should adopt when you are working with the laminar airflow. So these are the major textbook that is mentioned in your syllabus, dear students. So you can use any microbiology textbook, okay, in that particular matter. Okay, so thank you, dear students, for watching this particular class. If you are having any doubt